Hi, today I'm going to go through my feedback on your slides. So let's just get going. All right, so for this one over here, I think a more meaningful or informative title could just be LLISO. I mean, you've already said you're going to talk about the GE-based solution, and it'll be clear that you're providing an overview of it. You don't have to have that as part of the actual title. So, and then here, just to reduce the amount of words that you have on the slides, because you already would have LLISO up here, you can just say recombinant glucocerebrosidase. So that's the specific enzyme. So instead of saying just a recombinant enzyme used to treat Gaucher disease, if you just say recombinant glucocerebrosidase. Now, the previous people in your group will have already talked about glucocerebrosidase as the enzyme that is deficient. So that's just one. So that's just one point, and you can just make that be more specific than saying enzyme. And then the use to treat Gaucher disease. I mean, it should be pretty obvious. So I'm not sure it needs to be written on your slide. You can, but if you want to do that, put it on the second line. Actually, now that I say that, I think you should say used to treat type 1 Gaucher disease, because there are type 1, 2, and 3. We're focused on type 1 here. So this should be its own sent. So this here just means put it on a new line. If you have two different points you're making, put them on different lines so it's more obvious. Produced in genetically engineered carrot cells. ProCell X technology can be your third one. You don't need the periods at the end of the sentences because you're just going to have phrases, not full sentences. And anytime you can reduce some of the complexity, the visual complexity of the slide, you should. So get rid of anything you don't really need. And so I don't think you need the periods at the end there. You should have some sort of illustration. I mean, it's rather bland just to be confronted with a text full of slides. I mean, a slide full of text. So add some sort of illustration here. It could just be a box of LLISO. I mean, it doesn't have to be complicated, or it could be a reminder of, what this, I had this up for to show someone else in your groups, something like this, a pathway to show that in Gaucher disease, this enzyme is blocked. And so this is, producing that same enzyme again. Just something to help show what's going on in this example. Okay, over here, this is all covered by the person doing the background and problem, so you should not be covering it again. Okay, so that's already been done by your group. This would be repetitive at this point, so we don't need it. This is the same information on a different slide background, and I have to say I find this slide background rather terrible. This was painful to my eyes, like I was having trouble looking at it when I was trying to grade your slides. So please don't use this background. Okay, use something else. This background is fine. I'm not sure what it's trying to be, but it's better than this one anyway. Okay, so we don't need that one either. Again, just way too much text. And here, I think you're focusing on the production. So it could be production of recombinant glucocerebrosidase. And I mean, this is something you can say verbally. It's not important enough to write it on the slide. And then just say a gene for glucocerebrosidase in carrot cells. That's all I could find to keep here. And then you would explain this elsewhere. So what you're lacking in here, so you have very limited detail about these carrot cells and the genetic engineering, and you haven't even shown how they're grown. I mean, I think it's kind of cool. Like if you just do a search for LLISO bioreactor, you'll see these cool bioreactors that are orange because these are carrot root cells, right? So I think that's rather cool. And so being able to show how they grow these carrot cells in these disposable plastic bioreactors, and you can see them nice and orange here, that'd be great. And, I don't know, maybe one of these articles like this one, or I'm not sure, I didn't go read it myself, but it might have additional information for you, additional detail that you could put in here to help us understand this genetic modification, right? How did they get the gene into the cells in the first place? Is there an interesting promoter you could tell us about? Anything like that. Did they codon optimize it? I mean, I don't know the answers to these questions, but if you can find out anything, any amount of detail like that would be good. Illustrations to show what you're talking about would be good too. Carrot cells are grown in a bioreactor. Enzyme is extracted and purified. Those are the only words that need to be on the slide. Certainly could probably be combined with this as part of the production of recombinant glucocerebrosidase. So gene in the carrot cells, grown in a bioreactor, extracted and purified. Okay, and then that's where it'd be nice. Now you can show a picture of the bioreactor to go along with that. Over here, so FDA approved for treatment of type 1 Gaucher disease, more affordable treatment option. And then here, I was trying to think of like a more direct way of saying that, or something like demonstrated promise of plant-based, that's supposed to be plant-based pharmaceuticals, something like that. Anyway, if you can think of a way to say it with fewer words, that'd be great. And then this has all been covered. I mean, this is the point that you're making at the end here. So this is repetitive, or I guess this one over here. I guess these both are kind of the same point that you're trying to make here. And I think current impact and success, that's a great slide to make that point on. We don't need a whole nother slide to do that. All right, so things to work on, more detail about the genetic engineering and improved illustrations that actually are meaningful. Change the slide background, reduce the text by a lot. Okay, let me know if you have any questions about that.